Genetics influence health and disease as well as human traits and behavior. Researchers are just beginning to use genetic technology to unravel the genomic contribution to these different phenotypes. So I'm going to discuss about the application of genetic engineering. First is the application of genetic engineering to human genes. Growing advances make it increasingly likely that scientists will someday be able to genetically engineer humans to possess certain desired traits. Of course, this raises numerous ethical and legal questions. So one of the applications of genetic engineering to human genes is testing for traits unrelated to disease. Diagnostic testing supplies the technical ability to test pre-symptomatic at-risk individuals and/or carriers to determine whether they will develop a specific condition. Through advances in single-cell diagnostic and fertilization technology, embryos can now be created in vitro. Then only those embryos that are not affected by specific genetic illness can be selected and implanted in a woman's uterus. So let me first define in vitro. So in vitro is a procedure used to treat fertility or genetic problem and assist with the conception of a child. So this process is referred to as preimplantation genetic diagnosis. Preimplantation genetic diagnosis is a procedure used prior to implantation to help identify genetic defects within embryos created through in vitro fertilization to prevent certain diseases or disorder from being passed on to the child. The idea behind PGD is simple. Or blastomere is removed from an embryo that has been fertilized in vitro, and this cell is tested for genetic abnormalities. If the blastomere does not appear to have the defect under investigation, the rest of the embryo can then be transferred to the mother's uterus. So as you can see in this figure that the orange one are the affected embryos, while the green one are the unaffected embryos, which can only be transferred to the patient. This is building better athletes with gene doping. Trainers and athletes involve the use of performance-enhancing drugs that were originally meant to treat people. This practice is called gene doping. Gene doping is the transfer of genes or genetically modified cells into an individual as a potential method for illicitly enhancing athletic performance. It frequently involves such substances as erythropoietin, steroids, and growth hormone. Erythropoietin is a hormone secreted by the kidneys that increases the rate of production of red blood cells in response to falling levels of oxygen in the tissue. Steroids is an organic compound which include many hormones, alkaloids, and vitamins. Growth hormone secreted by the pituitary gland. Gene doping is defined as a non-therapeutic use of cells, genes, or genetic elements to enhance athletic performance. Lastly is creating designer babies, which referred to as creation of so-called designer babies. Baby is a baby that is the result of genetic screening or genetic modification. Embryos may be screened prior to implantation, or possibly gene therapy techniques could be used to create desired traits in the baby. Designer baby simply means to choose what baby you want. The traits that could be changed in a designer baby are its gender, appearance, intelligence, disease, and personality. This genetic technology has not yet been implemented. Nonetheless, it continues to bring up many heated ethical issues. Let's now move to the application of genetic engineering in animal genes. The goal of genetic engineering in animal genes is to make products to improve animals and to develop microorganisms for specific agriculture uses. is transgenic. Transgenic is the transferal of a specific gene from one organism to another. A transgenic animal is created once the second organism incorporates the new DNA into its own genetic. Take a look at this picture. They removed a DNA from the spider and inserted it to the goat, and then the goat produces a milk with a silk on it. So this is an example of a transgenic animal, which is a silk-producing goat. Gene splicing is used to introduce one or more genes of an organism into a second organism. In splicing, the donor DNA must be cut and pasted or recombined into a compatible fragment of DNA from a vector, an organism that can carry the donor DNA into the host. This process is used to produce transgenic animals. Scientists use reproductive cloning to produce multiple copies of mammals that are nearly identical copies of other animals. Cloning is the process of producing similar populations of genetically identical individuals that occur in nature. The first cloned animal, a sheep named Dolly, in 1996. Reproductive cloning begins with somatic cell nuclear transfer, then remove a nucleus from an egg cell and replace it with a nucleus from a donor adult somatic cell, which is any cell in the body except for an oocyte or sperm. For reproductive cloning, the embryo is implanted into a uterus of a surrogate female. Next is on plant genes. Genetic and genetic engineering has played an important role in improvement of plant production. These are the following applications of genetic engineering in plants. So first is hybridization. It is the crossing of different varieties of plant species. Hybridization is used to produce plants with desirable traits. Vegetative propagation is performed on hybrid plants. Plants with foreign DNA are called transgenic plants. So it has introduced many new characters in plants, and it helps a lot in improving the yield and quality of crops. Genetic engineering have developed a salt-tolerant plant, Arabidopsis. So it has the following steps. First, the scientists identified a gene coding for a channel proteins. These proteins transport sodium N11 across a vacuole membrane. Sodium ions are stored in a vacuole. Thus, sodium does not interfere into plant metabolism. Second is the scientists cloned the gene and use these genes in genetically engineered plants. These plants overproduce the channel protein. Then third, the modified plants grow well in water with a salty solution. Irrigation causes salinization of soil. It reduces crop yields. Today, crop production is reduced by 50% due to salinization. This problem is solved by producing salt-tolerant crops. Production of salt, drought, and cold-tolerant crops can increase the agricultural yield. Last but not the least is the application of genetic engineering in microbial genes. Microorganisms can be diverted from their original functions to be used by the industry for a wide range of applications. Microorganism as bioproduction system. Microorganisms are widely used to produce compounds not accessible by classical chemistry, such as proteins or antibodies, as well as existing chemical compounds. These are some examples of chemicals and biochemicals accessible by microbial manufacturing: lysosinic acid, linolenic acid, omega-3, and omega-6. Base biomolecules manufacturing. Genetically modified microorganisms are widely used for producing biomolecules. Examples of these are the antibodies, peptides, proteins, and enzymes. Microorganisms for bioremediation.
Microbial-based strategies are promising approaches to clean up soils from contaminated sites. Microorganisms used to perform the function of bioremediation are known as bioremediators. Some examples of contaminants that can be processed by bioremediators: the nitrate, phosphate, pesticides, cyanides, sulfur compound, rubber, and hydrocarbons. Biofuels manufacturing. Sustainable biofuels are essential to reduce the dependency on fossil fuels and limit our impact on the environment. So there are two major areas of research are lignocellulosic biofuels obtained by microbes and biofuels from algae. Bacteria-based biofuels. Microorganisms such as yeast can ferment the cellulose contained into the cell walls of plant waste to produce ethanol. This is also termed as lignocellulosic biofuels. Please, the microalgae-based biofuels. Microalgae such as Botryiococcus brownii, Dunaliella terliolecta, Pleurochrysis carterae could also be an alternative to fossil fuels as they produce lipids that can be processed into biodiesel. That's all about the application of genetic engineering.